Okay, it's the last lecture for Chapter 14. We're going to be looking at blood type and trying to make sense of how blood type works and how to prevent producing cross-reactions between a person's blood type and any of the blood cells or sera that may be administered to them. So here we go. So blood type is determined genetically. And there are 31 genes that are known to control the production of identifier proteins or identifier molecules on the surface of red blood cells. Most of these identifier molecules are proteins or glycoproteins. But the two that are most important, the two sets of genes that are most important, are the ABO genes and the RH gene. And again, expression of these antigens on the surface of the red blood cell is a result of heredity. So you inherit from your parents your blood type. So let's try to get in a little bit more and see what all this means. So if this is a red blood cell and you want to explain blood type, you've got to first understand what the surface antigens on the red blood cell are. So for type A, if you think about the gene sequences for the type ABO blood grouping, if a person has a particular set of sequences in the DNA, then they'll produce the A glycoprotein hanging off the surface of the red blood cells. So we refer to these as surface antigens. They're antigens because the immune system recognizes them and uses them to discriminate between self cells and non-self cells. So early in fetal development, the immune system takes an inventory and determines what are the identifier proteins I should expect to see associated with the cells of this person's body. And if it then encounters surface proteins or identifier molecules that it doesn't recognize from this early on inventory of the body, then the immune system will respond and do its best to remove that whatever cell or whatever component that it's discovered that bears those non-self surface antigens. Okay, so type A people produce the type A antigen on their blood. Type blood cells, type B people produce the B surface antigen. If you're AB, you produce both the A and the B antigens. Know that there is no such thing as an AB antigen. It's just the combination of expressing both B antigens and A antigens. Now here's the tricky part. If a person is type O, they don't have an O antigen. Instead, that means that they have no antigens. They have neither the A antigen nor the B antigen hanging off of their membranes. So that's one gene, but there's a second gene that's also important for blood typing, and that's the RH gene. If a person is Rh positive, that means they have this Rh gene hanging off of their, not gene, but Rh protein hanging off of their red blood cells. It's called Rh because it was first found in the rhesus monkey, Rhesus, and so that name has stuck here in the United States. In other countries, it's sometimes referred to as the D antigen, D as in dog. So, if you were in Britain, let's say, they might be talking about the D antigen hanging. But those are the same, they're just different names for the same surface protein. If you are Rh negative, then that means you don't have the Rh antigen hanging off of your red blood cells. So our blood types are a combination of the ABO gene and the Rh gene. So for a person who is AB positive, they would have the ACE, they would have B antigens, and they would have RH antigens, all found hanging off of their cell membranes. A person who is O negative has no surface antigens as determined by the ABO blood group and the RH blood group genes, has no antigens at all. So this is O negative. This person is AB positive. And of course, anywhere in between. If we were to look just at this A cell here, that would be an A negative cell. If we were to add some RHs here, then this would be a B positive cell. Okay, hopefully that makes sense. That's the antigens. Now we need to think about the antibodies. Antibodies are produced by cells of the immune system in order to protect the body from invasion by non-self cells. And these antibodies are found in the plasma of the blood, and they're also found throughout the body in any of the extracellular fluids. So if a person is a type A blood grouping, then the person would not make antibodies against the A antigen, because otherwise, if they did, then they would have antibodies out there grabbing a hold of every single red blood cell they could attach to. And then, because antibodies can hold multiple antigens or bind to multiple antigens, what we would see would be crazy clumps of red blood cells forming that would then lead to all sorts of circulatory disorders, not the least of which would be coagulation of the blood as it circulates in the veins, in the blood vessels. So a person who is type A, though, they don't express the B antigen. And so we would expect that they would have in their blood plasma anti-B antibodies. Every time my hand touches the screen, it gets weird like that. Stop it. Okay. So a person who's type A would have anti-B antibodies. A person who's type B would have anti-A antibodies, while a person who is type AB, because they've got both antigens, would have neither the anti-A or the anti-B antibody. So neither of them would be there. If they're type O, because they have no surface antigens in the ABO blood group, then they would have both anti-A and anti-B antibodies. Hopefully that makes good sense now. If a person is Rh positive, then they are not going to have antibodies against the Rh antigen. If a person is Rh negative, then they will have antibodies against it because it doesn't occur naturally in that person's body. So, if we were to then consider our AB, let's say somebody who is AB negative, they won't have anti-A because they got A. They won't have anti-B antibodies, but they will have anti-RH antibodies. Stop it. Because since they don't have the RH antigen, then they would make antibodies against it. So your job, one of your jobs on the test will be to tell me if, I give you a blood type, a person's blood type, you have to tell me to whom this person could donate their red blood cells. And you could also have to tell me what blood types this person might be able to receive red blood cells from. Okay, so hopefully this all now makes good sense. So let's talk for a moment about cross-reactions and then we'll be done. So cross-reactions occur when a person who's a person whose blood type is is given red blood cells that are incompatible to the antibodies that they produce in their body. Because antibodies are capable of binding to multiple antigens, when, let's say, we have an antibody here that binds to, maybe this is an anti-B antibody. And so it binds here to one of the B groups hanging off of this red blood cell. And over here it binds to the blood antigens on this red blood cell in the person's body. And pretty soon another antibody comes along, and it's capable of binding to this B and to this, the B on this red blood cell. Pretty soon you have this great big cluster of all wrapped together red blood cells, and that's going to cause some problems in the person's body, not the least of which would be sort of a massive coagulation event. So these circulating clumps of red blood cells may lodge into and plug up small blood vessels. Remember the circulating embolus, causing an embolism. And the results of this can be very painful and could also be fatal. This clumping, we refer to that as agglutination, just like we referred to the agglutination that occurs in, when we're doing blood typing. And we rely on that agglutination as a means of typing or ascertaining a person's blood type in order to make sure that we don't do any cross reactions. So there it is. Everything you wanted to know about blood, but may have been afraid to ask. If you've got more questions, please don't hesitate to reach out. You can email me. You can leave a message on my office phone. But if it's the weekend, you'll definitely want to email me. And study hard, and I'll see you next week.